தெரிஞ்சு ஸ்பீக்கர் ஆன்ல இருக்கு. பிரசன்னா ஃபோன் பண்ணா எடுக்கவே இல்லை. 2 டைம்ஸ் ஃபோன் பண்ணேன்.
Nishikant, good evening. Your voice is not audible.
Sir, you are in unmute, sir. Nishikant, sir.
Yes.
Okay.
Sorry,
Okay,
now it
sir.
is.
Good evening.
Good evening, sir.
How are you?
Yeah, fine.
The things are going for IPC.
Yes, it's very hectic for me.
Yeah. We will be coming. Actually, 3 staffs and some of us will come with you. We'll meet you. Sure.
Sir, shall we start?
Yes, you can start, madam.
Okay.
Nishikant, can we start?
Sir is in mute. Unmute, sir.
Yes. We can start now.
Then we start.
Okay. Okay, sir. Good evening to one and all. I warm welcome to our resource persons, Dr. Nishikant Raut, sir, Professor and Director of Maharaj Nagpur University. Welcome you, sir. And I would welcome to all the participants from this 5th day of Faculty Development Program in the title of Modern Approaches in Herbal Drug Development, conducting by Savita College of Pharmacy, Savita University from Tamil Nadu. It's organized by Department of Pharmacognosy, Savita Institute of Medical and Technical Science from Tamil Nadu. And what is the aim of faculty development program is to equipping teachers with skills and knowledge that are essential for inculcating ethical values in students and guiding and monitoring their progress towards entrepreneurial career. Hosting by Ms. Jupley, Associate Professor in the Department of Pharmacology, Savita University from Tamil Nadu. Now I would like to call upon our head of the department and professor of Dr. Thirumalai Kumaran sir to produce the achievements and rewards of our resource persons, and welcome you, sir.
Thank you, ma'am.
Okay,
Thank
sir.
you. Thank you, one and all. With the blessings of our Chancellor and Director, Vice Chancellor and our Registrar Madam, and also our Principal Sir, today we are going to end our day of 5 on the FDP. In this, I wanted to introduce my friend for the past 10 years. We were together working in a society for ethnopharmacology for the past 10 years. He's very close to me and is a able person for this session also. Nishikant is a professor in the Department of Pharmaceutical Sciences, Director, Department of Lifelong Learning and Extension, Rashtakant Doji Maharaj Nagpur University, Nagpur, and he is having 19 years of experience in teaching. and he completed his master's in pharmaceutical chemistry in 2002, and he has received his PhD degree in pharmaceutical science from RTM Nagpur University, where he is working now in the year 2010, and he has undergone postdoctoral fellowship from USA, University of Illinois at Chicago, under the clinical pharmacognosy section, and is very much interested in working in resveratrol and anthocyanin and in novel transgenic metachinases and development of novel drug developments in the osteoporosis research. His area of expertise includes natural products, medicinal plants, and phytopharmaceuticals. He has many papers in his credits in national and international journals, and he has he has worked in COVID time in the RTM Nagpur as a nodal officer in the diagnosis of COVID patients, and he has undergone 1,300,000 COVID-19 samples under the ICMR approved laboratory, and he has got an best teacher award, best researcher award in the year 2020 from RTM Nagpur University, and he is the one of the organizing person for IPC, which is going to be held in the January 2023, and he is actually a joint coordinator for Society for Ethnopharmacology Nagpur chapter, and also actively involved in various activities for the promotion of science and traditional medicines. Even though he is a pharmaceutical chemistry person, he is working in many plant drugs and novel drugs development from the plant sources. With these few credits of my friend Nisikant, I just want to have you for what do you call felicitation, madam.
Sir?
Jubilee, madam. Yeah,
Yes, sir.
felicitation, please.
Felicitate our today's on behalf of Savita College of Pharmacy. We would like to felicitate our today's speakers with the medicinal plants which they're planting in the name of yours, sirs.
Nishikant sir, on
Yeah.
behalf of you, we have planted a plant in our medicinal herbal garden
Thank you,
by
sir.
our students. This is the felicitation for you
Okay, thank
in
you,
online.
sir.
Okay.
Yeah, am I audible, no?
Yes, sir.
you are.
Audible, sir. Yes, sir.
So, shall I start?
Thank you, sir. Tirumulai Kumaran, sir. Thank you for your valuable information about our resource person, sir. Thank you, sir. Okay, sir. You can start, sir, now.
Okay. Yeah.
Okay, sir.
A very good evening, one and all.
Nishikant,
Yeah.
please make it full screen.
Yes, it is full screen.
No, sir. You are. Yeah, okay, sir. Yeah.
For you, it may be full. For the audience, it may not be. What about other audience, please? Others?
No, sir, it is not in full screen, sir.
Please, Nishikant, please make it full screen. Here, I just want to say one more. So, Nishikant, you make it a full screen. My student and my favorite student from Ramachandra, she joined every day. She is listening. Dr. Vinodini, are you there, madam? Vinodini, madam.
Is it full screen now?
Yes, sir.
Yeah.
Now screen came, sir.
No problem.
Okay, thank you.
Okay, sir.
Yeah, first of all, I would like to thank Kumaran sir for giving me opportunity to share my a small work here before the learned audience, because we know for this faculty development program, we have the regular faculties, the new faculty, budding faculties, we can say, and some of the research scholars, they also join this, and all of them are interested to gain something new from the faculty development program, to learn something new, and I'm also a learner, If I make any mistake, please pardon me. This is a small work which I did during my postdoctoral fellowship at University of Illinois at Chicago. That is a College of Pharmacy. That was established in 1869, and I got a very good experience there as a postdoc fellow under the able guidance of Dr. Gail Mahadi, my mentor. She is a motherly figure for me, and I would like to thank her for everything she gave me during this postdoctoral fellowship. So this is a small work that we did there. Actually, I have started this work during my postdoc. That was a project from NIH to Dr. Gail Mahadi, and I had also submitted that as my application to University Grants Commission for getting the postdoctoral fellowship under Raman postdoctoral fellowship scheme. So during that, we have worked on this transgenic Medaka fish. I got an opportunity to establish a complete setup for Medaka fish facility at University of Illinois under College of Pharmacy, under the clinical pharmacognostic division, which is headed by Dr. Gail Mahadi. And as I am a person of pharmaceutical chemistry, I was not having a single clue about what I'll be doing when I'll be working on the transgenic Medaka fish. Because the word transgenic I heard during my bachelor's. After that, when I took admission to pharmaceutical chemistry, so obviously we know that in chemistry we always deal with the certain structures and a lot of chemistry in that. And in that also, I was from the analysis side. So it was far difficult for me to understand, but I tried my level best to understand those things and started a very fundamental work there. And that was the wonderful experience that I'll share with you. So we know, if you can read this statement, one of the features which is thought to distinguish man from other animals is his desire to take medicines. So most of you might be what we can say. Do you agree with this statement? Most of you must be agree that this is the feature, that desire to take medicine is differentiating us from the animals. But I don't agree with this, because you might have seen several animals to eat a specific herbs. So for example, if you might have seen dogs eating some kind of grass or some kind of herb. So that particular dog gets it inheritedly, that if he has some GI problems, he has to eat something emetics, right? So this is not true in my sense. It is something different, that the whatever intellectual things we got, that is making us different from the other animals. Now we come directly to the drug, because the theme of this particular workshop is drug development through the natural products. So we have already learned this particular definition of drug, that a drug is substance used in the diagnosis, treatment, mitigation, or prevention of disease or disorder in human beings or animals, right? But we never consider that as that food, or exclusively a food as a medicine. But what our Ayurveda or traditional system of medicine tells us, we in Ayurveda the concept is the food thy medicine. That means food is our medicine. If we eat a proper diet or proper food, that is a medicine for us, because we know our food habits. It contains lots of herbs, lots of spices having medicinal properties. And if we observe the food pattern right from north to the south, that means we can say the Jammu Kashmir to the Kanyakumari, the food pattern goes on changes according to the environment those people experiencing, right? We are at the middle in Maharashtra. We found a mixed type of food habits, north as well as south. And if we go to south, again the food habits goes on changing, right? So these particular food habits, what we are having, it is a medicine for us if we take, because nowadays because of this fast food, the food habits are changing. Now, if we go towards the drug discovery and development process, so we always think that it is a very tedious process. When we were in our undergraduates, we go through the several things that particular drug discovery and development process, and you might have experienced that question, just how a drug discovered and developed. This is a common question usually being asked in several question papers in almost right from in all the colleges or university examinations. So how these drugs are discovered actually, and whether that is so simple that we can write 3-4 pages and yes, drug discovery is done. Is it so? Obviously no. So drug discovery is the process by which new drug candidate is discovered, and this is very simple statement that the new drug candidate is discovered, but it is a very complicated process. And as we have already studied, from the 10,000 compounds or 10,000 drugs, only one can reach to the market. And sometimes if it has certain adverse effects, if it shows certain adverse effects even after the launching into the market, with the post-marketing surveillance, if it is found unsuitable, so that can be removed from the market. So this particular process is very tedious and time consuming. Almost 10 to 15 years are required to establish a drug for a particular disease or disorder. So if you see the flow of the drug discovery and development, so sometimes we screen the set of compounds, or with the knowledge of certain receptors or a particular activity of a particular class of compounds, we try to design a particular molecule, that is called as the drug design process. And sometimes accidentally we come across a particular active compound and we consider that as a lead compound. Then we develop it further in order to enhance its biological activity or pharmacological activity, and to decrease the adverse effects, right? So for that, what we do, we try to optimize the structure. We go for a structure activity relationship kind of the studies, and that is also very tedious job nowadays with this computer aided drug designing and several softwares available. We can go for this in silico techniques to use for the structure optimization, and we get some scores, and depending on that we can shortlist some molecules. We can actually go for then a wet lab technique to synthesize them in the laboratory, and after that we can get the actual drug. And if that particular drug, it shows a proper activity as compared to the lead compound, it shows less toxicity as compared to the lead compound, proper bioavailability, and it is having appropriate metabolism. That means it is excreting from the body at a proper rate, and it is not accumulating. In that case, we can go for further studies for this in the process of drug development, right? Then if we screen the novel compounds, now we know that we have got several compounds from the nature, and the molecules which are coming into the market, those are also having some link with the natural compounds. And we know that in the recent past, the Nobel Prize was given for the discovery of the artemisinin or the isolation of the artemisinin to UU2, right? So it is having that much potential, and several compounds, they are also coming from the natural compounds, natural products. Now, the major advances which have been taken place in the natural product research is the isolation, purification, and chemical characterization. Now, initially, when we go for the traditional medicines, so usually those traditional medicines are usually given in the form of the crude drug, right, or some decoction or some extracts like that. Now, as the science grows, we were trying to understand what is there in the crude extract, what are the different active constituents in them, and we tried to isolate them, purify them, and then characterize them. Now, it allowed us to administer the controlled dose of that particular compound, and it also allowed us to administer the active compound or the herbal mixture alone, not with the other cofactors or the other compounds which are of no use as per the modern science, right? Now, but along with this, when we go for the isolation, we face many times these problems. If we go for the crude extract, so we know these crude extracts, they have a very complex structure, having number of phytoconstituents, and we don't know the exact number and what kind of compounds they are having. So we usually go for the qualitative test, what kind of or what is the nature of the compounds that a crude extract is having, and after that we try to isolate the active compound, and that is again a very time consuming process. So it requires, nowadays though we have sophisticated instruments with us, still it is a very time consuming process, and it requires several screens. If we go on fractionating the active compounds, we need to screen it periodically as we get it, and we have to go for several screens, and that is again a very time consuming and money consuming process. So now if we use partially purified, still they are having the number of compounds, but the complexity or the complex nature is somewhat decreased. Again, we can say the isolation is shortened, and sometimes during this partial purification, the particular active compound is being lost during the purification process, and it is not in the effective concentration in which it can show its pharmacological activity, right? So these are the problems, and sometimes some of the substances which have been lost during the purification process, those are responsible for the synergistic activity of the active phytoconstituent. So we call them as cofactors, and if these cofactors are missing, sometimes that particular compound either does not show the activity at all, or the activity seems to be decreased. Now, if we have a highly purified compound or the highly purified fraction, now the purification of individual substances from extract, again we know this is a time consuming process. It requires a very large amount of the starting material. Sometimes it is in kgs and we get it in milligrams, right? Sometimes we lost it completely. We isolate a completely wrong compound, and that is not having any activity, and it is of no use to us. Active substances originally at too low level to be purified, so that is again a problem, and it does not show its activity. And again, the cofactor is also important. If cofactors are missing, sometimes that particular, though we can isolate the active compound, it does not show its activity because of the absence of. Now, this is again a very important story which was published in Times of India. Now, one of that is also Dr. This G.J. Champanineri. So he was critically ill due to some reason. He was not knowing that. Actually, he was taking some anti-diabetic medicine of the herbal origin, considering the herbal drugs are safe for using it, but it was having lots of lead in that, and he was suffered from a lead poisoning. So it is not always that the herbal drugs or whatever medicine, herbal medicine, in the name of herbal medicines we are taking are safe for use, right? So if we think of the traditional medicines, so we always consider them as a safe, right? But to what extent they are safe? To what extent these traditional medicines are safe? Because if we use these traditional or folk medicines, we usually don't believe them, and as being the scientific fraternity, we don't believe them if they tell us that this particular herb or this particular drug is having a miraculous activity. We don't believe them because of the unavailability of the scientific evidences, right? Second thing, these things are true until those medicines are being used according to the traditional systems, the way in which they have been prescribed in the traditional literature or the traditional knowledge, right? But we don't want to take the risk for taking such medicines. Sometimes we believe, sometimes we don't, being this having some scientific knowledge with us, right? So when modern societies adopt such long-standing health practices outside of their traditional context, what does it mean that traditional context means? The traditional context here is the way in which that particular medicine is being prescribed to take along with some milk or hot water or any other thing. And in that case, if we are taking some Ayurvedic medicine or something like that, we need to follow certain pathya, apatya, and what to eat, what to not eat. The time is defined, at what time exactly you need to take that, blah, blah, blah. So there are several other things. And what we did, we have broke that particular chain of traditional medicines by applying our scientific knowledge. And as we applied the scientific knowledge to the traditional medicines, it has lost its traditional context, right? And those traditional medicines now become the complementary or non-conventional or alternative medicines, right? Because we have applied the half knowledge. We never try to understand what science is behind the way of taking that particular traditional medicine, right? Therefore, the extent to which the traditional use of an herb ensures that the corresponding herbal drug is safe. However, this may be a matter of debate. So we should follow those practices which are given in the traditional literature, and we need to follow that exactly what is being prescribed, right? If we try to modulate that, obviously it will be no longer a traditional medicine, but it will be a complementary or a non-conventional or alternative medicines, right? And they may produce their adverse effects then. So if we want to establish the safety and efficacy, so obviously these things are already known to you, that we go for a preclinical studies, and if we undergo preclinical studies very successfully, then we go for a phase 1 clinical trials, then phase 2, phase 3, phase 4, and we still monitor the drug, right? So I'll not go much detail in that. But in preclinical studies, what usually we do? We usually go for the pharmacological profile of that particular drug, and we study them on either isolated cells or organs. Then we study receptor binding characteristics. So with the modern context, we study that particular medicine very well, but there are certain hurdles in that. in some of the pharmacological activities, we lack the good animal models of human disease, or either they have there are very few models available, or even in some cases the human disease models are not existing. The research is going on that to develop certain models also, right? So it becomes very difficult to predict the potential therapeutic use of certain drugs. Now, in preclinical studies, we also study the pharmacokinetics. We also study the toxicological effects of those drugs. Then this is again a very important slide. Mechanism matters, but what does it mean? Now, here is one of the example which was published in Nature Medicine, in which the Pfizer and one more company, that is Medivision, they were working on one drug that is Dimebon, and for the Alzheimer's disease. And suddenly they found that in the phase 3 clinical trial, the drug was failed. It was not showing the significant pharmacological activity or therapeutic efficacy as it was predicted during the preclinical studies, as well as in phase 1 and phase 2 clinical trials. And they had to shut down the trials, and obviously that is a big setback for any company after investing such a large amount, because we know that millions of rupees or the dollars are being put in to develop a single molecule. And this is really a very bad setback for any company to shut the clinical trials, right? So the problem is because this FDA does not require any understanding of the mechanism. When we though they require certain data related to the clinical trials or the preclinical investigations, but they never enter into the mechanism of action of a particular drug, that how the drug acts, right? So gaining a clear understanding of how a drug works before it enters into clinical trials will be the intelligent route, because it can save your time as well as the money. So what are different advantages of understanding the mechanism of drug action? So most drugs produce their effects by binding to a protein molecules. Drug binding often leads to a conformational change in the protein, and we know that particular process, how it so we study that ligand receptor interaction on all those things, and then it is all this data is being compiled, and we propose a mechanism or possible mechanism that this particular drug acts through this particular pathway. And sometimes that particular mechanism also fails, and we don't know how exactly drug is working. So knowledge of drugs mechanism of action enables us to better dosing through monitoring of the drug's effect on target pathway in the patient, to help stratify clinical trials, to focus them on those patients most likely to respond, and to predict its side effects or adverse effects, what we consider. Now, this mechanistic knowledge is certainly worthwhile before entering into uncertain world of clinical trials, because we know at even after the phase 4 clinical trial, that means the post marketing surveillance is again very important, and some of the drugs, they have been called back from the market because of certain adverse effects and because of certain adverse things being noted, right? So this is a case study. We have tried to establish the mechanism of the dietary anthocyanins from the natural sources in the osteoporotic model, that is being comprising of the transgenic Medaka fish, in which we tried to test our dietary anthocyanins into these transgenic Medaka models of osteoporosis. Now, osteoporosis, again, I'll skip this because osteoporosis is well known to you. So, now what is present treatment that is available for the osteoporosis? So, obviously, this intermittent parathyroid hormone injections are available. Those improves this bone strength and enhance osteoblast differentiation and regulates calcium levels in the blood, and biphosphonates. So, those inhibits the activity of bone resorbing cells. But they do not enhance the bone formation, but they are associated with the several adverse reactions or adverse effects, and they usually fail to address the pathophysiology mechanisms of in the bone remodeling. But what is the significance of this study? What we did? So what we found that there is a positive link between the high consumption of the dietary flavonoids and anthocyanins and indices of the bone health. But the mechanism by which they produce these beneficial effects in the bone development or ameliorating the osteoporosis in the experimental animals, that was totally unclear, and not much literature was available because I did this study in 2016-17. So that time it was quite unclear. So we set the hypothesis that these dietary anthocyanins, they increase the osteoblastogenesis and reduce the osteoclastogenesis in human osteoblasts as well as in transgenic Medaka models of osteoporosis. So we use two different transgenic models, the double transgenic and triple transgenic models. What are these double and triple transgenic that we'll see in the subsequent slides. Now, as we know, these anthocyanidins or anthocyanins, these are water-soluble colored pigments obtained from different sources. You can see on the slide, usually this blackberry is most famous for obtaining the anthocyanins or anthocyanidins. Now, the models which we used during our study was the cultured human HFOB osteoblast and this teleost fish, that is Japanese Medaka. Those are almost transparent. What are the advantages of using these fish model? These are the models or the animals which are having which enables us to go for the quantitative high resolution imaging. Also the bone development similar to the mammals. So in mammals, the bones is usually directly developed from the mesenchymal condensation, and therefore it is quite similar to the mammals. Then the genetic networks is well known, which are controlling the bone formation, and these are highly conserved in vertebrates. It is not changing with time, and because of that, this particular model is very suitable for utilizing for this particular study. And the most important is the transparent. So we can combine powerful genetics with the high resolution imaging, and we could observe the bone development at periodically in this particular transgenic Medaka fishes. Now, again, why we have chosen this particular Medaka? So the optical clarity allows live imaging of the bone cell behavior at high resolution in intact specimen. So we could mount them in a specific Petri plate in which we could mount it when the fish is alive, and we could go for the high resolution imaging with the fluorescent type of microscope and all those things. And this also allows to monitor the proliferation, migration and differentiation of the osteoblast and osteoclast in living embryos as well as their interaction with each other. Now this again in vivo imaging of this SP7 Osterix amcheri transgenic fish allows us to quantitatively determine osteoblast differentiation, as we could measure the fluorescence, intensity of fluorescence with the bone mineralization as well as the bone resorption. So when bone develops through this osteoblast, so these are certain the expression of the genes which are responsible for this, and it is usually started from the mesenchymal progenitor cells, which get converted into osteoblast progenitor, wherein the collagen 10 alpha 1 is being expressed, and then osteric gene is expressed when it forms a premature osteoblast, and under this osterics again it gets converted into the mature osteoblast. And in case of osteoclasts, again the macrophages or monocytes, from macrophages and monocytes it develops into the osteoclast progenitor, and under the control of this rankle expression it gets converted into osteoclast, and then it gets converted into for the mature multinucleate osteoclast for the bone resorption. Now, what is this double transgenic metacalines? So these in this particular double transgenic metacalines, there is a highly restricted expression in the skeletal regions. So mCherry under control of this sp7 osteric promoter genes, we can it usually controls it, and we can restrict a particular skeletal regions in which we want to study. Then so transgenes sorry so these are present in mature and mineralizing osteoblast, and they are usually involved in the notochordal sheath mineralization, and but are required for perichordal ossification. So you can see the mCherry or that particular the colored pigment, or what we can say the fluorescent protein is being tagged along with the osterics, and because of that we can have the fluorescence, we can get the fluorescence in the bone, and because of that we can quantify the bone development also, right? Then second transgenic medaca, it is also called as the col10 simply. It expresses the this nuclear lamina green fluorescent protein under the control of col10 alpha 1 promoter, and the cells appear prior to the mineralization of the notochordal sheath. So this is the initial stage of the development, what we have seen on the first slide of that development of the osteoblast. So during this, even these cells, they appear prior to the mineralization. That means before the bone development actually. So these cells remain on the outer surface of the caudal centra. And this is triple transgenic. So it is usually tagged with three different fluorescent proteins, SINE, GFP, and mCherry also. So during this, that is this receptor activator of NF-kappa B ligand, that is we call it rankle, the regulator of the osteoclast formation and activation. And here one more protein is being tagged, that is called as the heat shock proteins. So before start of the study, the larvae that is on the 3 days post hatching are being compared with the heat shock and without heat shock. And if after heat shock, it shows the SINE fluorescence or blue fluorescence, we can say. In that case, we take those larvae for the further study. So that heat shock protein, it is bidirectional, we can say. That means with it can go towards the CFP or GFP. Depends how we proceed, right? So its overexpression, this rankle's overexpression induces osteoclast stimulation and osteoporotic bone loss. So it expresses rankle together with the CFP under control of the bidirectional heat shock promoter, and green fluorescent protein under the control of cathepsin K promoter in osteoclasts. Now you can see in these images, can you see the cursor? So first one is the image stained with the alizarin, and that is the skeletal matrix we can see. And the second one is the cathepsin K, with under the control of cathepsin K, this NLGFP expression in the head and tail, right? And in the third image, we can see the distribution of Osterix mcheri, positive premature osteoblasts in the cranium, vertebral columns, and caudal fins. In the D, that means here we can see the neural arches and the centra at this particular development stage, and this is a stacked image in which we can see the osteoclasts, those are in green, and this osteoblasts also, which are in red color. So F is also the alizarin strain skeletal matrix. We can see it very well here, and this I and J, they are also the stack images, where we can see the development of the this green fluorescent protein, or which reflects the generation of the osteoclasts after heat shock. So these are again very clear images about these the confocal images of the double and triple transgenic metaka for the visualization of osteoblast and the osteoclast during osteoporotic conditions. So first one is of the osteoblast, in which Osterix mcheri is being expressed, and we can see a very well developed bones. The second image is the mineralized matrix, and it also shows the presence of osteoclast. And third one is the after heat shock. That is, it induces the resorption in the bone cells. So D and E images are plain alizarin red staining of the mineralized matrix. This is a wild type medaka, in which we can see clearly the neural arches, vertebral body, and humeral arches very clearly. Whereas in the heat shocked or the mineralized matrix, after the expression of the rankle, we can see that resorption or this demineralization in the bone very clearly, right? So what methods we have used? So in this we have used two different methods. We also use the cell lines for our study in order to understand the different modes in human osteoblast. We have used human osteoblast 1.19 cells. Those were obtained from ATCC Manassas, and were cultured in 1:1 mixture of hams, F12 medium, Dalberg's modified eagle medium, and we have treated with some of the extracts, as well as resveratrol and other anthocyanins. We have also used the double and triple transgenic medakas in our study, and these larvae at 9 to 12 days post fertilization were staged and then treated for 5 days with the extracts and compounds from 1 to 40 micrograms per apple. And the osteoblast and osteoclast formation and distribution were analyzed using fluorescence and confocal microscopy, and the bone mineralization was visualized by alizarin red and calcium. So this was the method in which these human osteoblasts were treated with increasing concentration of the anthocyanins and resveratrol, and growth was assessed using cell titer glow. And obviously here also the fluorescence was measured in that. In vivo methodology, what we did, so these medaka embryos, those were hatched at 6 days post fertilization. They were screened for fluorescence. Just remember those blue fluorescence under CFP after heat shock, and those larvae were only taken for further study. So these hatch larvae were fed for 5 days with blackcurrant extract, delphinidine, delphinoglucoside, resveratrol, and other anthocyanins. And at 5 days post treatment, these were analyzed for alterations in the fluorescence and bone mineralization. So this was the mounting plate. You can see this small tiny Medaka larvae, it is approximately 0.5 to 0.6 mm in length. It was mounted there, and then it was screened for the fluorescence. So, and again the confocal microscopy. So, what results we obtain in osteoblast, human osteoblast? So, resveratrol and anthocyanins, they increase the osteoblast proliferation in human osteoblast cells, and they reduce the apoptosis at 1 to 10 microgram per ml. And again, this when these cells were serum starved, so usually the serum starvation induces apoptosis in human osteoblast, and with the increasing concentrations, we could found that the apoptosis process has been restricted by the anthocyanins. Again, the qPCR results, they have shown that there is a marked differentiation in the expression of various genes, in which we could found this as the different backs to BCL2 ratio. It shows that there is a restriction in the apoptosis process. So P53 usually it regulates the cell division process. So that regulation was also being reduced in the cells. Then HDAC1, it is responsible for the osteogenic differentiation. Those are also being reduced, and PGC1 alpha and other SIRT1 and those things. So, so in the double transgenic Medaka treated with this resveratrol and other anthocyanins, they showed the enhanced osteotic gene expression, indicating increased osteoblast growth and differentiation. Right. So it is directly related with the intensity of fluorescence. So fluorescence was found to be increased with resveratrol. So we can clearly, even visibly, we can say that in the control, if we see this particular region or this particular region or this particular region and compare it with the treated group in resveratrol, we can see the well-developed bone matrix here. Again, this resveratrol treatment in triple transgenic Medaka reduces the bone loss induced by Rankel induction. Right. So here we can compare directly with this and with this. Here also we went on the detailed study of this. So resveratrol was found to be improved the bone recovery, and it reduces the resorption in cold-10 as well as the Rankel models of the transgenic Medaka fish. That means the double and triple transgenic models of the Medaka fish. Sorry. So this resveratrol, it has also found to decrease the bone resorption after the heat shock induction of the Rankel HSCCF in Medaka larvae. So we can see directly in the control. We have, that means the untreated and without heat shock group. We can see the neural arches, the vertebral body of centra, and the hemal arches also. And wherein this heat shock Rankel induction process, we can see the demineralization here. And with the treatment, the condition has been somewhat improved with the 2.5 microgram per ml, and with 2.5 microgram per ml, it is quite improved. So we can say that it is reversing that resorption process. So in summary, we can say that in transgenic Medaka, these anthocyanins were more active than the resveratrol in some of the cases, that they were effective at 5 to 10 fold lower doses. And this particular work suggests that anthocyanins likely to have multiple mechanisms of actions. That means it is not acting through a single mechanism. So it can inhibit the HDAC1 in cultured human osteoblast. They can increase the SIRT1 to 1 and 3, and PGC1 alpha expression, and acted as a P53 antagonist so as to reduce the apoptosis process. So in SP7 Austerix Mcheri transgenic medaca, both our models, molecules, punidine, thioglucoside, and resveratrol, increase SP7 Austerix and Runx to expression along with the osteoblast proliferation and differentiation. And in transgenic Colton or Rankel medaca, all the anthocyanins reduce the Rankel-induced bone resorption, of which the most active was the punidine thioglucoside. This was the paper. It was published in 2020, having impact factor 7.65 right now in phytotherapy research. And my mentor was so gentle to me that she asked me to be a corresponding author, as I was sharing this first authorship with this Zitauren, who was my colleague during my postdoctoral fellowship. She's from China, and Dr. Simon Lee is her mentor. And my other teammates, Temito Plowell and Sheetal Patel. So we could publish that, and more results and more papers are in pipeline on this particular work. And the further extension of this particular work is again under process, and I'm still in touch with them, and I can get the manuscript for my suggestions and these things. So the further extension is the transcriptomic and ingenuity pathway analysis, that is called as IP analysis. And there are several other labs which are being involved in this particular study. So this UIC Tech interns, the Karen, Nankela, Alice, and Paulina. So they're from the Monterrey, this Mexico. They were also visiting fellow to the UIC. Then UIC Research Informatics Core, that lab is also involved in our study. Dr. Kanabar is there, and Dr. Mark, they were coordinating for that. And we are also using the core genomic facility at UIC with this Dr. Zarema and Nina. And at Department of Pharmacy Practice, Dr. Temito Plowell and Dr. Sheetal Vin Patel, they are coordinating for this particular work. So we are expecting the results. The work is still ongoing. We are not having the complete data with us right now. Some of the data we got, but it is under the interpretation stage. So I'm not presenting it here. And presently the RNA sequence analysis for mRNA is going on. So obviously with the results of these studies, it will give a clear picture or the understanding how exactly these anthocyanins are working for the bone development or decreasing the osteoporotic conditions. So the possible mechanism that was being proposed by us in that particular paper was that the resveratrol and anthocyanins, they stimulate the SIRT1. So it decreases the osteoclastogenesis. Again, the SIRT1 also acts antagonizing this cathepsin K, Rankel, and NF-kappa B, and SIRT1 also stimulates the Runx or Austerix or SP7 genes, and thereby increase the osteoblastogenesis. So, so these studies establish Medaca as a novel in vivo model for drug screening for the osteoporotic condition and as a bone modulating compounds. So this is just a take-home message that gaining a clear understanding of how a drug works before it enters clinical trials is the intelligent route of drug discovery and could increase the likelihood of the drug success. And most of the traditional medicines derived from medicinal plants possess excellent pharmacological activities, but they lack scientific evidences. And nowadays, these scientific evidences are very, very important because the Western world, they want for more data, the clear picture, how your drug is acting, blah, blah, blah. and the generation of this scientific evidences for the effectiveness of traditionally used medicinal plants will be more safe and can be considered as evidence-based traditional medicines. We are also working with here with this developer that is Go Vigyan Anusandhan Kendra, and we are undertaking some of the work on the Go Grita, the Grita preparations which are there in the Ayurvedic formulations. We are trying to validate the activities as well as other things related to the Grita formulations of the Go Vigyan Anusandhan Kendra. I would like to acknowledge and show my immense gratitude to my mentor, Dr. Gil Mahadi, who was supported with the R21 grant from NIH, that is under this National Center for Complementary and Integrative Health. The University Grants Commission for awarding me a Raman Postdoctoral Fellowship, and Schlumberger Foundation, who awarded a postdoctoral scholarship to Dr. Temito Plawal, who is from Nigeria, University of Ibadan, and again this UIC Tech seed funding to Luna and Dr. Mahadi. Bioinformatics analysis in the project. This particular work was performed by the UIC Research Informatics Core, and that was supported by NCATS through this particular grant. So I thank all of them, and I thank my teammates. This is me, Dr. Temito Plawal from Nigeria, Dr. Sheetal Patel, and my mentor and motherly figure for me, Dr. Gail Mahadi, and this is Dr. Jita Rain. We used to call her Ada, and we used to call Dr. Lawal as Funmi. Anyway, so I would like to take this opportunity to introduce 72nd Indian Pharmaceutical Congress. This was basically being awarded to us in 2020, but because of this COVID pandemic, we could not make it, and we are able to make it now in January, during 20 to 22nd January 2023. All of you are welcome to this event. These are some of the highlights. So you know what is 72nd IPC, or you know this Indian Pharmaceutical Congress, what it is exactly. Every year it is being organized, and this time Indian Hospital Pharmacists Association will be hosting it, along with the Department of Pharmaceutical Sciences, Rashtrasinda Tukadoji Maharaj Nagpur University, Nagpur. This is our university. We are celebrating a centenary year. In 2023, we'll be completing the 100 years of glorious history to the Nagpur University, and department is quite old. It was established in 1956, and several stalwarts have been graduated from our department, and they have their own industries, and our alumni are very strong, as from the Dr. P.P. Somani, who is also our alumnus, and who is right now the Drug Controller General of India, and who will be the president for this Indian Pharmaceutical Congress Association. And this time the theme which was proposed was the access to quality and affordable medical products. These are some of the highlights. That first time the CEO conclave, HR conclave, medical devices, blockchain technologies, artificial intelligence, several other things will be first time introduced during the Indian Pharmaceutical Congress. Apart from that, there will be several other parallel sessions, and the scientific content will be very, very high. So I urge you to register for this event. The registration fees is very less as compared to the 71st Indian Pharmaceutical Congress, which was held in 2019. Though the prices have been increased 1.5 to 2 times in several domains, still we tried to keep the registration fees on lower ends. For students, it is 3,000 plus GST, and for teachers, it is 5,000 for GST. The early bird registration that was up to 30th November has been extended till 15th of December. So all of you are welcome to Nagpur for this particular conference. Accommodations are also available. We already booked several hotels and other places of accommodation where we can accommodate the students, the delegates, faculty members, and the dignitaries. What you will get the exclusive kit bag. It will also have attractive gifts in it. The feelers given by certain pharma industries. Almost 15 companies have agreed for CEO conclave. That means the 15 big people will be there for CEO conclave, and these CEOs, we are planning for the interaction with the students also. That means they can showcase their journey from their student life to the current positions as a CEO or the managing director of some big companies. The registration fees will also include the breakfast, lunch, dinner, high teas, free access to exhibitions, entertainment, and in entertainment we are having this. Can you guess? Have you gone through our website ipcnagpur.com? Javed Ali, Pushpa Fame, Sri Valli Frame. Javed Ali will be the attraction for entertainment for youngsters, and on second day we'll be having Nitin Mukesh for life, right? So there will be a free entry to world-class musical fountain, which is recently being opened for the public, and you are also having the students are also having the opportunity to work as volunteers. Then we'll be having exhibitions and several companies, as as I have just now told that 15 companies have agreed for the CEO conclave. Obviously, these big companies they will be having their exhibitions during the exhibition session at the exhibition hall. These are the places of interest for visiting, so you can have tourism here. Several, and Nagpur is known for tigers as well as the oranges, and you can enjoy the oranges during the conference. So we are trying to keep it free for the delegates. Only oranges, not the tigers. And these are silent features, and the call for abstract is open till 25th December. So you are also invited to submit the your research work for presentation at IPC. So with this, these are the account details and registration links are there. So we can directly jump to the registration link from here. So if you want, I can give you a short tour to the website if Kumaran sir allows me to do so, because I know I have exceeded time limit. Kumaran sir,
Yes.
Kumaran
You
sir?
can open, no problem.
Okay, thank you, sir. So this is IPC www.ipcnagpur.com. The highlights are also given on the first page, and you can click register here and you can get directly on the registration page, right? You can go for registration. Online registration is available, and these are the rates for registration which I have already shown. So go for the IPCA. These are different 5 bodies of which the IPCA is made up of. The LOC, this is local organizing committee, and our just 1 minute. So in patrons, we have Honorable Minister for State, Honorable Shri Nitinji Gadkari, as well as our Honorable Chief Minister and Deputy Chief Ministers, and Sanjeevji Rathore, Minister for Food and Drug Administration. They are our chief patrons. Then our Vice Chancellor and other administrative. Then we have a local organizing committee under the leadership of Shri Atulji Mandlekar, who is Assistant Commissioner FDA Maharashtra, as well as the Officer on Special Duty for Honorable Minister Shri Nitinji Gadkari. This is also a well-known face to you, Dr. Milinu Mekar, who is currently the president for APTI, and as our organizing secretary, and second one, Dr. Prakash Itinkar, organizing secretary for this event, and myself as a treasurer for this IPC. So this is again a kind request to all of you. Please register for Indian Pharmaceutical Congress, and you are welcome to the Nagpur. Thank you.
Thank you, sir. Thank you for your wonderful and valuable speech about transgenic Medaka models of osteoporosis. Thank you, sir. Now it's time to discussion of today's
Yes,
session.
please.
Participant, please kindly raise your doubts.
Sir, good evening, sir.
Good evening, ma'am.
I am Dr. J. Kumari from Wales University. Our presentation excellent. Sir, I have two doubts. One is nowadays there are many researches going on regarding the traditional medicinal plants. Usually, before discovery into a drug molecule, we are going the extraction followed by fractionation. Then you go for bioguided fraction by in vitro study. When you
Right.
come to in vitro studies, there are very difficulty to do the work based upon the activity or based upon its molecular mechanisms in any lab. Can you suggest, sir, what we have to go and proceed with that one? Because without in vitro study, also we cannot find out the mechanism of the particular fraction or whatever may be. What is your suggestion regarding that one?
So best thing is to collaborate. I'm also working with different labs in US as well as the other nations. So the best thing is whatever we don't have, we can get it through collaborations. So collaborative projects, they will not only give you the facilities in those labs, but it will also give you to interact and to learn several other things. So if you can able to collaborate with someone, it is best.
Okay,
Because
sir.
we cannot hire everything, or we cannot afford
Yes,
the
sir, yes, sir.
outsourcing for such kind of studies.
Yes, sir. Yes, sir. That one more doubt, sir. We are isolated we are doing the many research work in the traditional medicinal plant. Previously, we did only extraction. Then recently, we did that bio-guided fraction. Then after that, which is a more worthy, sir? Whether the extract or only fractions or isolated compound, which is more potent when
Now, ma'am,
compared?
it depends.
Okay.
As I have shown three different slides: the crude extract, partially purified, and highly purified.
Okay.
So it depends how you perform your fractionation or the isolation process. And as you are telling that you have gone for the bioactivity guided fractionation,
Yes, sir. Yes, sir.
right? So if
Yes.
you are getting the synergistic activity or the more the potentiation in the activity,
Yes,
so
sir.
obviously you got that particular lead in that.
Okay, sir. Okay, sir. Okay, sir. Thank you, sir. Thank you,
Thank
sir.
you,
Thank
ma'am.
you, sir. Thanks, information.
Thank you, ma'am.
Good.
Nishikant, I have a doubt.
Yeah, sure, sir. Please.
You have given a transgenic method for osteoporosis.
Mhm.
How would it be related to human body osteoporosis?
Actually, the bone development in Medaka, particularly this transgenic Medaka fish, particularly Medaka fish, the bone development is through the mesenchymal condensation. That is a very similar process that is in mammals. And therefore, we directly correlate with the human beings. And in order to understand that, we have parallelly run those compounds or screen those compounds in human osteoblasts in cell lines.
Cell lines, okay.
Yeah.
Because zebrafish model is people are doing, but that is not correlating with the human body. That is why I have a doubt on this.
The
Correct?
models which are available, the either using the rat or chicken, and but those involve surgical interventions. And this particular model, it is not having any surgical interventions.
That's why. That's what correct. What you're saying is correct. But all the mammals, they are not really. Most probably, all are not same. No. So you're
Yes,
saying.
though all are not same, but the basic mechanism of this bone development is same. So that's why we can correlate directly.
Okay. Since you have done in cell culture, then we can accept it. No problem.
Yes.
Um, any other people, please, madam, Jubilee, madam.
Yes, sir.
There is a question in the chat box. Just read, madam. In the chat box. Before that, who is KHR channel? Yes, raised. I think so.
Sorry.
Please, Anand Vandarkar.
Anand.
Please.
Hello, sir. Good evening.
Yes, good evening.
Sir, you have carried out the work on resveratrol and anthocyanidine for osteoporosis.
Mhm.
But the resveratrol, being a BCS class 2 drug, which is having a very poor solubility in water, how have you managed to put into the particularly the fish so that the embryos can take up the particular resveratrol?
Okay. So this resveratrol, they were dissolved using DMSO. A very small amount of DMSO, and we could dissolve it. So we are having the ready-made solution with us, and we could give it in. If you could observe, so we kept these medacal larvae in those wells, having a particular solution in which that particular solution was miscible. So that we have already done, tried, and then only we have used that.
Sir, but the anthocyanidine being a polyphenolic compounds, and they have a very poor bioavailability in human beings.
Right.
So because of that, we are going for liposome or phytosomal kind of the formulations.
Right.
How you address this issue of bioavailability?
Yes, obviously, I'm also working on this bioavailability issue or solubility enhancement, and that is my apart from this herbal drugs, I'm also working on that. So this particular issue can be addressed with the different techniques of the solubility enhancement, and thereby we can try to enhance the bioavailabilities. And if we can take them in natural forms, so in natural forms they are very much bioavailable. Hello.
Hello.
Thank you, sir. Thank you.
Okay, thank you, sir. Sir, next one participant asked opinion, sir. That's kindly express your view on identifying the pharmacodynamism of a natural bioactive compound from natural source with all hurdles, restrictions in the drug development process. Here, either conventional pharmacological approach or reverse pharmacological approach is optional for successful drug development, sir. He asked opinion, sir, for this. That is
Yes.
Karthikeyan and Ramadas, sir.
Okay. Is it in chat box? So yes.
Yes, sir.
there
Chat
is
box only, sir.
Okay. Of a natural bioactive natural source. This pharmacodynamic dynamism of the natural bioactive compounds from natural sources, obviously we need to follow that the development process, and we need to follow that. We cannot directly go for this pharmacological approach or reverse pharmacological approach. We need to go through what are being prescribed by the regulatory agencies if we want to develop that into a particular therapeutic agent.
Okay, thank you, sir. Sir, shall we go to end of section, sir? Any
Yes, ma'am.
participants raise? Shall we go, sir?
Yes, please, ma'am.
Yes. Next, I can invite Dr. Prasanna Bharati, ma'am, Assistant Professor and Faculty of Savita College of Pharmacy, to propose a vote of thanks. Prasanna Bharati ma'am.
Is there?
Ma'am, am I audible?
Yes, yes.
Yes, ma'am. Yes, ma'am. You can proceed.
Okay. Good evening, ladies and gentlemen. I'm Dr. S. Prasanna Bharati. Deem it a great honor and privilege to propose the vote of thanks on this final day of the online faculty development program hosted by the Department of Pharmacognosy from Savitha College of Pharmacy. Let me first of all start by giving glory to the Almighty God for making this event a resounding success. First and foremost, I thank all our speakers and today's guest, who despite their busy schedule has contributed the valuable knowledge in pharma to our listeners and with a keen interest and responsibility. We are grateful for the guidance you have placed on us, sir, especially to Dr. Rishikant Rauts of today's session. Also, I thank our principal, Dr. Ramayan Verumurugan, and Vice Principal, Mr. Dinesh Kumar, for their unstinted support for this 5-long-day program. We are grateful to Dr. Thirumalai Kumaran, Head of Pharmacognosy Department, for his hard work to ensure this event as an informative and most memorable occasion. My heartfelt thanks to the head of various departments and faculties for their continuous support and encouragement in all our efforts. We owe special gratitude to all the distinguished invitees who were present during the entire session, accepting our invitation and evincing interest in especially covering this faculty development program meeting, and to all the good hearts who work behind the screen. So, last but not the least, finally, I leave you with this inspiring quote by Martin Luther King. An individual has not started living until he can rise above the narrow confines of his individualistic concerns to broader concerns of all humanity. So, once again, I thank you all for making this session a most fruitful one. So, here goes the ending of the session. Sir, Thirumalai Kumaran, sir.
Ma'am,
Yes, ma'am.
thank you.
Yes, thank you, ma'am.
Thank you, ma'am. Finally, I just want to say a vote of thanks to everyone for the ending of session. First of all, I wanted to say thanks to my teacher, who is she is the head of Department of Pharmacognosy, Wales University, Dr. Jaikumari ma'am. She is my mentor when I was studying BPharm in KM College of Pharmacy, and she is the guiding force for me in many ways also. So thank you, ma'am. Jaikumari madam, thank you, ma'am. And I have to say thanks to our Chancellor, sir, Director, sir, and guiding forces of our university, Vice Chancellor, sir, Registrar, madam, our principal, and all the faculties who supported me in successful conduct of this program from the day one to today, and all the some of our students also helped me in many of the way, particularly Kirtikeshwaran from 3rd B Pharmacy. He was hosting his YouTube, our official YouTube. He is doing that work on that. And I wanted to say thanks to him also. And the other students who are all involved and helped me, and my non-teaching staffs also, who has helped me in preparing the certificates to each and everybody who has attended the workshop, FDP. And finally, I should say thank only my right and left hand, my madam, Miss Vandana. She was helping me from the day one, and before that also till now. I wish to thank her also, and she is the one handling everything. Even I had a problem in day before yesterday. She was the handling all the things. So she is saying that it is new to me, sir, and she was saying about complete success. So I wish to thanks to Vandana madam and other people who are attended this program all the 5 days, and all the speakers who are all my friends and guiding forces. Like from day one, I started with Prakash Itankar, Dr. Prakash Itankar. Day 2, Dr. Rajan sir. Day 3, Dr. Syed from Jamia Ahmedabad University. Day 4, Dr. Sivakumar, who is my friend and brother. And day 5, my friend Dr. Nishikant Raut. these two people, Prakash Itangar and Nishikant, even though they are having a busy schedule on conducting this, organizing IPC, they helped me in successful conduct of this 5 days event, FDP. Thank you all, Nishikant and Prakash. Very
Thank
much
you,
thankful to both of you, and we'll make a success in your IPC also. Thank you.
sir. Welcome.
Thank you, one and
Welcome
all.
to Nagpur.
Thank you, one and all who are participated all over India, who have registered to my FDP. And
Sir, Kumaran sir,
please.
we are also hosting Indian Science Congress,
Thank
108th
you.
Indian Science Congress.
When it may be.
And I am a convener for that Children's Science Congress.
Oh,
That
that
work is also going
I've
on.
seen it. Yeah, that I've seen it. Anyway, you are working for that also. In that issue schedule, you helped me to success of this FDP. Thank you, one and all.
Thank you.
And my teacher again, Jaikumari Madam, who is also a guiding force to me. Thank you, Madam. Thank you all. And with this, we are winding the session. And last information is that please fill the
Feedback, sir.
feedback form.
It is in a chat box only.
It is there in the chat box. It will be only through that only we can identify who is the attended, and those people only will be getting the certificate. Please fill the feedback form. That will be helpful for us. And any area suggestions if you want to give, like Dr. Anand Bandrakar and my madam, Jaikumari madam, and other people. If you want, you can say, and you can put in the chat box also. Please. Anand sir. Dr. Anand sir. Are you there? Okay. Anyway.
Yes, sir. Tell me,
If
sir.
you want to give any suggestion also, we can improve, sir.
Yeah. No, sir. The session were very good, very informative. They have presented their core research work, and they have told us to go for the collaboration, which is a need of the hour.
Of course, you can contact me. I'll help you for collaboration, and even to collect the plant. I can help you through the Dr. Rajan sir, who is there in Ooty. I can.
Yes, sir.
help you.
Okay.
You can
Dr.
contact-
Anand,
Yeah.
where are you from?
Sir, I am from Goa College of Pharmacy. I am an associate professor in Goa College of Pharmacy, Department of Pharmacognosy.
Okay, your colleague to
Shailendra Gurau.
College. Okay, I'm coming for this World Ayurveda Congress.
Yes, sir. I am there in scientific committee, sir.
Okay, so
You are
definitely.
welcome. You are most welcome.
Yeah, it's nice interaction with you. Thank you.
Yes. Thank you, sir. Thank you very much. See you in Goa, sir.
Yeah, sure.
Thank you.
Sir, please share the mail ID and the contact numbers of all the speakers so that we can have that collaborative work or some plan material and all. We can get it from them.
Surely. We'll also do it with you people. We can also do collaboration. Dr. Anand sir.
Yes, sir. Sure.
Yes, no problem. Thank you.
Thank
Any
you, sir.
other
Thank you.
people who wants to give any suggestions? Any other people? Malapudi madam?
Sir, good evening, sir.
Madam.
First of all, I would like to thank my Savita University for giving us this opportunity. And thank you so much. The time was very apt to attend the FDP. We learned a lot of things through the FDP and arrange many more like this. We will always cooperate with you. Thank you so much, sir. It was very nice to hear the novel concept, whatever is happening in pharmacognosy. Thank you, sir.
Sangeeta madam? Vinodini madam? From Ramachandra? If you want to give any suggestion, I can improve, please. Anybody is there? Any other people? Doctor, who is that? I don't know. Sir, somebody, please, can you give me any suggestion, please? I can improve you. I can improve. Anybody, please? Okay. Then I can say proudly, I have completed my FDP successfully without any break. Thank you. Thank you one and all. Jubilee madam, thank you madam.
Yes, yes sir. Thank you, sir.
Thank you. Thank you all. Okay. We'll,
Thank you, sir.
yes.
Thank you.
Deliraj sir, yes. Thank you. Sorry.
Okay. No issues, sir.
Fill up the feedback form, all of you, please. Okay. Jubilee madam, can we convene the FDP?
Sir, I guess she has left the meeting.
Left. Okay.
Okay.
Okay, madam.
Thank you. Thank you, sir.
Thank you. Yes, thank you. Thank you all. We can leave. You will be getting the certificate shortly. Punegodi sir.